As you'll see from the accreditation on my opening slide there, I work in the Women's Health Academic Centre, and as such we are interested in all of the common complications of pregnancy, and not just in terms of pregnancy outcome, but in terms of the long-term cardiovascular and metabolic health of the offspring. And that's really where my interest has come from. I'm a cardiovascular physiologist by training. But if you're interested in the etiology of a disease, and if you want to actually look at the origins, you have to go, in this case, back to the womb, which is how I ended up effectively in an obstetrics department. So what I'm going to be doing today is to take you through what was Chapter 8 of the Task Force report written by Lucilla Poston. I'm going to take you through the sort of key points and hopefully illustrate that with some examples from the literature, and finish up with some mechanisms derived from the animal models. This, I guess, is the scale of the problem. We have an exponentially increasing epidemic of obesity, not just in the UK but across the world. And it's not just in adult obesity. We see that in terms of children, the number of obese children in the UK has tripled between 1990 and 2010. And these data from the States just show you that sort of exponential rise in the rates of obesity from one generation to the next. And the key point here is that this has happened too quickly for this to be explained in terms of simple Mendelian genetics. Genes just don't move through a population that quickly. So this is likely to be an environmental effect. We know from twin studies that childhood obesity has a heritability of about 50%, and there have been a number of common obesity genes which have been identified. Obesity is likely to be a polygenic disorder. But of the common variants that have been identified, which include FTO gene, the leptin variants in the leptin receptor, and other receptors in the melanocortin system, which are involved in appetite and satiety, the currently known common genetic variants fail to predict childhood obesity in birth cohorts. And so it's likely that childhood obesity as a disease represents an interaction between genetic susceptibility and environmental insults, such as nutrition in early life. And the first evidence that we had for a nutritional effect in terms of long-term cardiovascular and metabolic health came from the early Barker studies. So in particular in the Hertfordshire cohort here, meticulous midwifery records from the turn of the century with details about birth weight and maternal status allowed follow-up of the cohort some 60 years later. One of the clearest observations made was that low birth weight was associated with abdominal obesity in adult life. Subsequently, other cohorts went on to demonstrate that low birth weight or a disproportionate fetal growth was associated with other aspects of metabolic syndrome, and that was encapsulated within the Barker hypothesis, suggesting that an adverse in utero environment affects growth and development in early life, resulting in subsequent disease. And then that was superseded, I guess, by the theoretical construct, which was the thrifty phenotype hypothesis, which suggests that the adaptations that the fetus makes in utero to undernutrition or an adverse nutritional environment, whilst adaptive in terms of immediate survival, can have long-term implications for our future health, especially if the nutritional plane changes postnatally. And one example of that, the Dutch hunger winter, where there was a sustained famine which was imposed on the northern ports of Holland. So there was an embargo in reprisal for Dutch resistance, and this resulted in rations being cut to between 40 and 800 calories per day. Detailed clinical records again allowed follow-up, and what was observed was that those individuals that were exposed to the famine in mid or late gestation had impaired glucose tolerance, whereas the early exposure was more associated, most importantly, with obesity. And for the first time, this was shown to be independent of birth weight. So this suggests that the critical periods, which was alluded to earlier, are important in terms of the timing of a particular nutritional insult. So here's a schematic just showing you the effects of a potential mismatch between the in utero and the postnatal environment. An abnormal or undernutrition environment effectively changes the nutritional supply via the placenta and can result in impaired fetal nutrition. And then the fetal adaptations, or the plasticity of the developing fetus, makes adaptive changes through possibly epigenetic processes to adjust to that nutritional environment. Now, that nutritional environment remains the same postnatally as normal disease risk profile, but if the subsequent plane of nutrition increases, a high nutritional postnatal environment that can result in obesity. So other potential determinants of childhood obesity include rapid infant growth or catch-up. And we know that small for gestational age babies that show rapid postnatal growth over the first 12 months have an increased risk of obesity. And this has been shown in numerous studies now. And a recent meta-analysis suggested that there was a 2 to 3-fold increase in obesity and overweight in those individuals that crossed growth centiles in the first 2 years. So that's really highlighting the importance of a stable growth trajectory postnatally. And other studies have suggested that the catch-up growth, even in the first 8 days of life, may be critical to subsequent development of obesity. And of course, that implicates feeding practices. That study was carried out in formula-fed infants. In summary, the human cohorts provide some evidence for an independent association of birth weight with later risk of obesity. It's likely that the size of this effect is quite moderate. There was a recent systematic review in The Lancet suggesting that birth weight alone only had quite a modest impact on obesity. But historical cohorts also support the association between undernutrition during critical periods of development and subsequent risk. And if we combine the birth weight relationship with neonatal growth rates, that gives a much more robust relationship between birth weight and risk of childhood obesity. Again, highlighting the importance of neonatal growth trajectories and, intriguingly, offering the possibility of intervening to optimize nutrition and growth during that period. There's evidence to suggest that breastfeeding may be protective against childhood obesity. Although, again, the relative benefits of this may be somewhat have been overestimated. Various systematic reviews have put the beneficial effect between 22 and 7%, and the recent WHO data suggested a 22% reduction in overweight also. And the beneficial effects of breastfeeding are likely to arise through the reduced intake, just through the logistics of feeding, the slower weight gain that's observed in breastfed babies. So the possibility of milk-borne hormones in the milk, such as leptin and insulin, which may not be present in formula feeds. So obviously the composition of formula is critical to growth. And there's evidence that historically the formula has been quite high in protein, and recent studies, not least through the Early Nutrition Project, through Bert Koletzko, have done randomized controlled trials to look at the effects of the lower protein content in formula-fed milk, and they've found that compared to higher protein, there's an increased benefit in terms of childhood obesity, and that with the lower protein, the risk was similar to that observed in breast milk. So here's just a schematic showing you the potential effects of accelerated weight gain. A high intake of growth-enhancing nutrients, particularly protein in the context of formula-fed milk, leads to a high plasma and tissue levels of insulinogenic amino acids, and they're likely to drive increased secretion of insulin and insulin-like growth factors. And insulin is the key adipogenic hormone, so that will promote adiposity and contribute to weight gain. They conspire to increase to the long-term risk of obesity in the child, which can be tracked through into adulthood. So what about maternal diabetes, and what about the other end of the birth weight spectrum? We know that the baby on the left there, which is born small and shows rapid catch-up growth, is at increased risk of obesity. But we also know that the macrosomic baby on the right, which is probably born to a woman with gestational diabetes, is also not only heavier and fatter, but at greater risk of developing childhood obesity. And there seems to be this U-shaped relationship between birth weight and risk of obesity, which has been borne out in a number of systematic reviews, which generally show a positive relationship between increasing BMI and childhood obesity. And we know that babies are getting bigger. This is data from the Swedish Birth Registry from 1992 to 2001. Took account of nearly 900,000 pregnancies, and they observed a 23% increase in large for gestational age babies, which they attributed statistically to a similar 25 to 36% increase in maternal BMI. So larger ladies are having larger babies, and this was also associated with a reduction in smoking, which also can reduce birth weight. There's the potential, therefore, that increasing BMI in women of reproductive age and in pregnant women results in increased glucose, insulin, and leptin exposure into the fetal compartment, and this can result in this overnutrition of the fetus results in fetal macrosomia and can lead to permanent changes in the energy balance not least in the appetite regulatory centers in the hypothalamus and in energy homeostasis. And so this can result in, as we say, increased risk of childhood and adult obesity. And we know that in the UK, approximately a quarter of men and women were classified as obese in 2008. And in the UK, women are now the most obese in Europe. And approximately, although the numbers are slightly sketchy, it's approximately 20% of pregnant women are obese at the time of registering for primary care. So this is potentially becoming a huge problem. Obesity is the single biggest obstetric risk factor currently. And that's the importance of obesity in pregnancy was reflected in the publication of the recent CMACE and RCOG guidelines on the management of women with obesity in pregnancy. And here's just some data from the Motherwell birth cohort, basically showing that both maternal BMI in early pregnancy and gestational weight gain both have an influence on offspring adiposity. So the data here shows in panel A, percentage body fat in individuals that are stratified according to mother's BMI in early pregnancy. And within that stratification, there's also a significant effect of gestational weight gain within those BMI categories. And similarly for waist circumference, we see an increased waist circumference or risk of obesity with increasing BMI in pregnancy. In a more recent study, this was the Jerusalem cohort study published towards the end of last year. And again, they found a significant and independent effect of both pre-pregnancy BMI and gestational weight gain in pregnancy. And you can see that the effect of the pre-pregnancy BMI, which is the dotted line there, is greater than the effect of gestational weight gain alone. Gestational weight gain is actually probably a poor indicator of adiposity or indeed fetal weight, especially in obese women. Some studies have found no association between gestational weight gain and childhood BMI. The pre-pregnancy BMI is generally a better determinant of pregnancy outcome, although the association with gestational weight gain may be stronger in underweight mothers. But with in obesity, the gestational weight gain is very variable. It reflects not only the weight of the fetus and the placenta, but the amniotic fluid, but also, besides maternal weight gain, there's volume expansion in the cardiovascular system, there's water retention. So it's a very variable and perhaps not very predictive marker of adiposity in pregnancy. And in terms of in the UK, the evidence base is not really established for adopting the guidelines that were recently introduced in the States by the Institute of Medicine. And here they predicted what would be acceptable weight gains within the different BMI categories. And you can see that as obesity increases, as BMI increases, the acceptable margins for gestational weight gain are reduced. But the current UK position, as reflected in a consensus statement which was published last year in Pediatric Research, that was the result of a workshop in Brussels. They decided that the evidence was not there in a UK population. So as a measure of gestational weight gain, it was too variable, not sufficiently predictive. And these recommendations are all based on observation studies rather than any intervention to control gestational weight gain. So there are a number of randomized controlled trials which are looking at exactly that. And so I think the UK policy will await the outcome of those trials. It would appear, just in summary, that an appropriate pre-pregnancy BMI and perhaps an appropriate weight gain in pregnancy could be protective for childhood obesity. But as I say, the reports so far are of an observational nature, and therefore they are subject to residual confounding. The shared genetic and environmental characteristics between the mother and the child could obviously account for these relationships, but there does seem to be an effect of intrauterine mechanisms, and that includes the increased exposure to nutrients such as glucose, amino acids and free fatty acids from mother to fetus, which can affect appetite regulation and neuroendocrine functioning. But as I say, until interventions in pregnancy can be shown to reduce risk of obesity in the child, there can be no evidence-based translation to the improvement of child obesity. And this schematic just shows you the various potential determinants of childhood obesity. We have increased maternal BMI, gestational weight gain, and diet will impact not only on birth weight, but also in pregnancy on the risk of obesity. Maternal smoking, which I haven't touched on, is a major risk factor for childhood obesity, although the mechanisms are not clear. Probably involves chronic inflammation and placental dysfunction. But these prenatal factors all converge on a background of genetic predisposition, I would say. And then there are other factors such as diet and physical activity in childhood, demographic and social factors which will impact. And in the infant, there's an effect of breastfeeding, of accelerated growth, and also of sleep duration, which is intriguing, because any of you with children would argue as to whether sleep duration in babies was a modifiable risk factor, I'm sure, but I'd struggle. So what about interventions to reduce offspring obesity? Theoretically, it's possible that by reducing low birth weight, that could have a positive outcome on obesity, although studies that have tried to influence birth weight through nutrition have found that to be a particularly refractory outcome. Reducing postnatal weight gain through the promotion of breastfeeding is perhaps more amenable, but more amenable to intervention. The only randomized controlled trial that I'm aware of was in Belarus, where infants were randomized to either breastfeeding or formula feeding. However, childhood adiposity in that study was only a secondary outcome, and so it wasn't really powered sufficiently and didn't actually pick up an effect of childhood obesity. And as has been mentioned earlier, there are probably ethical and practical obstacles to further randomized controlled trials of breastfeeding. But the adoption of the International Child Growth Standards, which takes the breastfed child as the normative reference, will result in a lot more infants being classed as overweight or obese, and therefore potentially alerting healthcare professionals to early weight gain. Formula feed composition, as I mentioned earlier, the low versus high protein formula in the first year of life results in infants that had a lower BMI that was not different from breastfed babies. So the improved composition of formula feed, the inclusion of more accurate micronutrient content, and even probiotics, as we heard this morning, makes the formula feed composition more like breast milk and may in fact reduce in the long term childhood obesity. But again, we await the long-term follow-up of studies of this nature. So what about interventions in obese pregnant women? To date, there's no randomized controlled trial which has reported on the effects of weight loss or normal care in pregnancy on childhood obesity. And of course, there are inherent risks associated with weight loss in terms of conception and pregnancy. And current guidelines from the UK suggest that weight should be maintained in pregnancy. There is some evidence, however, for a beneficial effect of weight loss prior to pregnancy. There are increasing number of morbidly obese women who are undergoing bariatric surgery for weight loss. And remarkably, if you compare babies born to these women before and after weight loss surgery, there's a threefold reduction in the prevalence of obesity in siblings. And so that mitigates against any genetic influence and suggests a firm in utero mechanism which is giving rise to offspring obesity. And here's just a redrawing of that data. There was a 10-fold reduction in macrosomia in children born after bariatric surgery compared to their siblings born prior to, and a threefold reduction in obesity, again, before and after surgery. And associated with that reduction in obesity, there was also improved metabolic profiling. That's all well and good, but how do we address the potential mechanisms which could be involved in the programming of obesity, and why use animal models for this purpose? Well, mice, rats, and humans all share all but 1% of each other's genes, and of course we're all mammals. The advantage of using mice certainly is that you can use genetically identical strains, so you know that any interventions in pregnancy are purely environmental and have nothing to do with genetic traits within the animal. The relatively quick life cycle, especially in rodents, means that you can look at the effects of a dietary intervention in pregnancy on adult outcomes in a meaningful time frame. You can tightly control the environment, so you can reduce a lot of the residual confounding that can plague large human studies. And you can test, obviously, diets, which might not be ethical to test in human populations. And you can obviously look at different periods of development, different critical periods, which would be difficult again to achieve in humans. And there have been a number of animal models which have been designed ostensibly to test the theories that were thrown up by the early epidemiological studies. So a number of studies have looked at undernutrition to try and mimic the famine studies, a whole host of different interventions ranging from specific restrictions in protein to global restrictions of various kinds, all can result in an obese offspring. And if we compare, that's just an example. This animal was born to an animal which was subjected to undernutrition during pregnancy. The offspring have an increased fat mass. They're hyperphagic, which means that they eat more from birth, and they were disinclined to exercise when given the opportunity. So this has been termed a sort of couch potato phenotype, if you like. They eat more and they are disinclined to exercise as a result of mum having undernutrition during pregnancy. And in terms of overnutrition, there are various models which can affect the nutrient supply, both prenatally and postnatally. And again, they all have a similar effect in producing an obese rodent. So you can reduce the litter size of suckling rodents, which effectively is a model of neonatal overfeeding with increased milk availability. Diabetes can be induced experimentally during pregnancy, and you can see the effects in terms of adiposity in the offspring. And then various dietary interventions have been employed, looking at diets which are rich in fat, sugar, or generally an obesogenic diet. And that was an area in which we developed a model of rodent obesity at King's. So in this model, both rats and mice were given a highly palatable obesogenic diet for 6 weeks prior to and throughout gestation and lactation. And then we were able to follow up the offspring to look at their phenotype. So the only intervention was in the mother by giving them a fat, sweet diet. And the first thing we noticed in the offspring of these animals was that they ate more. So almost from weaning, within the first few weeks of life, you could detect an increase in the calorific intake of those animals that were born to obese mothers. They weren't overtly obese, they're kind of chubby here, as you can see on the right, but they have approximately a doubling in their fat mass, and if they are subsequently exposed to an obesogenic diet, then that weight takes off. So we wondered what was driving the hyperphagia, the increased appetite. And one of the ways to test appetite is to give a leptin challenge. And leptin, as you know, is secreted by fat cells proportionally to your fat stores and the size of your fat cells. And it signals to your brain to stop eating and that your fat stores are full. So the normal response if you give an animal a shot of leptin is that it will inhibit their feeding. And that's what you can see in the far panel there, in the offspring of controls. Leptin reduces 24-hour food intake, and that results in a comparative change in body weight over the same period. But when you give the same challenge to offspring of an obese animal, there's no effect of leptin. Their appetite and their body weight is completely refractory to the leptin challenge. So these animals, at 30 days of age, this is before they've become obese, they are already leptin resistant, and that is normally a state which is acquired chronically in obesity, but it appears that through exposure to maternal obesity, these animals are already leptin resistant. And what we noticed was when we looked at the leptin profiles in the neonates was that whereas in controls there's a normal leptin surge which happens over the first few weeks postnatally, in the offspring of the obese animals they're exposed to much higher levels of leptin over that period. And we were able to experimentally impose a similar leptin level in normal animals by giving daily injections of leptin, and in so doing we could actually recapitulate the leptin resistant phenotype. So again, in a leptin challenge when at 30 days of age, and also later on at 5 months of age, the animals that had seen the very high leptin in early life were and remained leptin resistant. So there are a large number of developmental programming mechanisms, which I don't have time to go into, just in case you were thinking that this was all a female problem. There is some evidence that a father's nutritional legacy can impact on the offspring function. So this was a study from Margaret Morris's lab in Sydney, published in Nature, and they showed that rats that were fed a high-fat diet became glucose intolerant and resistant as fathers, but the female offspring also showed impaired glucose tolerance at 6 and 12 months, with changes in expression of genes related to islet function. And similar changes in methylation status. So this suggests that a paternal high-fat diet can potentially alter the epigenome of offspring, in this case in a sex-specific manner. And this was just to show you that you can get multi-generational exposure through an environmental exposure in the F0 female there, and that can be transferred through to the F2 generation, ostensibly because that F2 rat or F2 mouse was an egg in the F0 female. So you can have an environmental effect, somatostatic effect on tissue development there. For it to go to the F3 generation, that requires a transgenerational phenotype, which means that the programming, the epigenetic programming through nutrition is occurring through the germline. And this recent study in Nature suggests that that can not only happen in females, but it can happen in males as well. So just to conclude, the key points that were raised in Chapter 8 of the report regarding determinants of childhood obesity include the nutritional status during critical periods of early development, that growth retardation in utero, especially associated with rapid catch-up growth, is a major risk factor for childhood obesity. There may be a protective effect of breastfeeding, although that may have been in comparison to higher protein feeds, and the relative effects may well fall as more studies come through. But the excessive fuel availability associated with maternal diabetes and obesity is also a major risk factor for childhood obesity, and a potential modifiable risk factor in terms of the better management of obesity and pregnancy. And animal studies have been invaluable in providing irrefutable evidence for the effects of maternal obesity in developmental programming of offspring obesity. But the absence of randomized controlled trials in humans really is awaited before there can be any firm public health policy interventions. And in terms of the recommendations which were made, the Institute of Medicine guidelines on gestational weight gain have not been adopted in the UK. And again, we await confirmation from randomized controlled trials that intervention has a positive effect on childhood outcomes. Low protein formula feeds may may prevent accelerated neonatal weight gain, but as I say, follow-up is required. And the results of dietary and lifestyle interventions in obese pregnancy are currently underway, and their results are eagerly awaited, not least the UPBEAT study, which is being run out of King's. Professor Poston is the PI on that, and this is a complex interventional study which is looking at the effects of dietary advice, low glycemic index, and physical activity, not just in terms of primary outcomes of pregnancy, but we also intend to follow up the children from UPBEAT and look at the direct consequences of intervention on childhood obesity. Thank you for your attention.